with us and others in the group. We came up to changing his biomarker from more of a responsive biomarker to a diagnostic biomarker, which will hopefully then lead him into a trial. So I think that he's making great progress on that as well. And then Richard Sky has his project is a pilot utility design clinical trial using salicylate for Alzheimer's disease. And this is already funded, so he's already gotten funding for this from his institution. And we had a discussion that if he increased his sample size slightly with additional funding, he could look at it as a utility design with a clinical endpoint. So then we switched to specific aims, so that specific aim 1 would actually, if he could get this additional supplemental funding, could be a utility design to look at functional testing. The second aim would be to look at the safety profile and feasibility, tolerability. Third aim was to look at PK and also biological effect using established Alzheimer's disease biomarkers. All right, and then he had an exploratory fourth aim, which is very interesting, to look at some the effect on some known Alzheimer's outcomes otherwise, as well as some new biomarkers. So I said anything wrong, guys? I'm good? We're good. It was a good group. We really enjoyed working and continue to work. Wow.
All right, so group 3, we had Wendy Galper, Pooja Khatri, and myself as the leaders of the group, and then Heather Fullerton, Waldo Herrera, Cindy Hsu, and Debbie Maddock as the mentees. There was a lot of bad "Where's Waldo?" jokes. Waldo has assured us that he's heard every single one. Yeah, so Waldo's doing his project is coenzyme Q10 for cardiac arrest and global ischemia protection. So he's looking to do an adaptive dose finding design. So we said, Waldo, you'll need a statistician. He said, well, great, I'm coming to Iowa. So, okay. Heather Fullerton, actually, one of the big things that happened this week in particular that I thought had a big impact was the office hours that we all had and all the confusion that they introduced. And Heather had put it in a great way. She says it was actually a really good thing to go through because she thinks she would have gone through it anyways. And here she went through it back to back to back to back. And you actually got a really good sense for what that was like as a whole. And so I think actually it was a really positive experience. For Heather in particular, her project is corticosteroids and acyclovir for prevention of pediatric stroke. And what we really struggled with before the class was the actual, the specific question we wanted to address. And I think we got there this week, but now we're dealing with some sort of feasibility issues as far as the imaging endpoint that we want to use for this trial. Talked about Waldo. Cindy Hsu. She has a project called The Role of High-Dose Valproic Acid in Out-of-Hospital Cardiac Arrest Neuroprotection. For this one, we actually got to a point where we thought maybe there should be another non-clinical study, so in a large animal model. So this week has still been really useful because we've been thinking about, okay, once that's done, what's the next study? What's the clinical study? And so a lot of effort has gone into that. Let's see. And then Debbie Maddock, her project is fiber optic endoscopic evaluation of swallowing to guide early extubation in the neuro ICU. And that one, we haven't really made any design changes this week, but I think she's received a lot of really good input as far as like operationalizing such a trial. So this week's also Group 4.
All right, Group 4. No hand signs. Please, you get it. Sorry. It's like half. Yeah, insider joke. Sorry. Yeah, you just sent the one. So Will yesterday said microphone to the faculty and the trainees should listen. So we decided to give the word back to our trainees and ask them to give us a couple of sentences, and we'll read them aloud for you. That's what we're going to do. So we had a great group, 4 participants in our group, Robin, myself, and Kurt were the group leaders, and I'm going to start with Lauren, my fellow intramural NIH neuroimmunologist. And Lauren had the following study, and I'll read it out for you. Developing a phase 1 study of pembrolizumab, a monoclonal PD-1 inhibitor in a clinically stable biologically controlled HIV-infected cohort on antiretroviral therapy using HIV-specific CD8 T-cell responses to identify a CNS reservoir. As CSF CD8 T-cell cytokine responses are not known following PD-1 therapy in HIV-infected patients, the pre-drug phase of the trial will measure the intra-subject variability of the CSF cytokine response, determine the feasibility of using the biomarker outcome in a planned trial. If the cutoff is met, the trial will proceed to a one-time dose of 2 milligrams per kilogram pembrolizumab and a post-drug cytokine response in the CSF. Within subject comparison will be used to compare the response in the CSF to the peripheral cytokine T-cell response to determine if there's a localized CNS response against HIV, indicating the presence of the viral reservoir. This will then be used to inform larger trials with the goal of achieving the sterile cure. So I think that's a pretty cool biomarker-driven study here. You want to do next?
So I think that we're making a lot of progress, and we also got Jeff Saber to join our group because he was in town, so that was a nice boost for all of us. So now I'd like to talk about Araya, who went on the study. Araya has an unusual enthusiasm. I really am grateful to her for all her energy and extra things she did to help them and the group in general. So her study is tolerability and activity of resulting for pain in type 2 myotonic dystrophy. The challenge here is a very rare disease, plus she's looking at pain. So we know that widespread pain is a well-known symptom of myotonic dystrophy, and the pathophysiology is uncertain. So she's proposing a phase 2 randomized double-blind placebo-controlled study. Now I'll tell you, she worked with a lot of statisticians. Similar to Heather, she had a lot of different opinions. And I think she's decided on a certain design, but I won't mention what it is because it can change. But we think that she's decided on a crossover, which is usually not very common, but because of the rarity of the disease.
There's more office hours today if she wants to talk to Bill about statisticians.
But I want to say that we were looking to have Kurt Feely in our group. He is a super statistician, and she also spoke to Jason Connor. Got a lot of great feedback from Bryson Sulla and Laura Dutton. So thank you very, very much.
And for what it's worth, you can talk to the same statistician again and potentially get another opinion.
So anyway, again, she's looking at safety, tolerability, EMG myotonia score, muscle strength, 6-minute walk test, lower limb time function test, depression score, patient-reported outcomes for quality of life, and inflammatory biomarkers. She has a great collaboration with a podiatrist at our institution who's very well funded. So that's positive too.
All right, next is Prasha. Prasha is an emergency doc in Stanford, and she is she submitted a project that already got through 1 round of reviews in grants, and this is a community-based, basically educational program, and here's to Prasha. The primary objective of the study is to assess the effect of an educational intervention on pre-hospital recognition of stroke using a step wedge design. The major progress includes refining my primary outcome and designing an analytical plan. So we had lots of back and forth in terms of primary outcome, analytical plan, who is the subject in the study, the EMT or the patient themselves. Primary outcome was changed from sensitivity, defined as the proportion of subject with a pre-hospital provider impression of stroke among those with a hospital-based diagnosis of stroke, to a utility outcome that combines the desired sensitivity threshold with the desired specificity threshold to assess the extent of over triage with higher sensitivity. So really, thank you. That was good. So basically, this is a prospective design. And so the prospective design was changed to a step wedge design, to include the control population in response to her reviewer comments. And they will now be able to compare the baseline phase and the post-intervention phase in groups 1 and 2. And I think a meeting with Kurt kind of solved the problem here. So don't go back to them, you might get another opinion, so stick with that. So she was able to maybe work with Kurt here to solve this issue. And yeah, I'm really excited to see what this goes.
I can tell you her, talk to people from FastNet, and then Jeff was here, so that was a really great opportunity. So thanks so much for all your hard work. And the fourth student is Kathleen Payne. Kathleen, are you still here? Not sure she is. So Kathleen has a great expectation, and she came and with all her energy and is proposing a study of coagulation cascade and acute stroke and post-intervention. And we were able to move her from a phase 3 trial to a pilot biomarker study, which was really important. And thanks to Will Moyer, who worked with her in advance, and I think she made progress here as well with Kurt Wheeling. And she basically is going to look at different coagulation cascade factors, and this will be tested before and after treatment for acute ischemic stroke, and that will be either with IPTPA with thrombectomy, IATPA with thrombectomy, or TPA alone. And also, she's looking at MR reperfusion and modified Rankin scores. So she is going to do a prospective observational study and great progress as well. So thank you, everyone, and I think the course is fantastic.
Our group wasn't the only the only lazy group that had people write things down for us. So again, Erica, I think, had most of the good ideas and did all the hard work, so I got tasked with reading these. So I feel like I contributed. So I think we had a great group. I think, I learned in graduate school that I can get a good course. You learn as much from your classmates as you do from your advisors. I think we had a lot of good discussions within our group and trainees giving one another good ideas. So it was a great group to work with. I think behind this is hard. My favorite quote that I read from grad school was one of my advisors saying, the reason we call it search and not research is because nobody gets to make the first time. So as we're all, I think, I saw some deer in the headlight moments, including in my office hours, when people realize this is really challenging. That's okay. Anything worth doing well is worth doing poorly in the first time. Or others. But so Francis Panosian, who moved from Rochester to Kingston, Ontario, is that right? Oh, okay, other way around. All right, going in the right direction. All right, from Kingston to Rochester during this, it was working on magnetic resonance spectroscopy as a biomarker. Magnetic resonance spectroscopy of the muscles of limbs will be used as a surrogate marker for disease progression inherited neuropathies, otherwise known as Charcot-Marie Tooth disease. The rationale and background for the study, in addition to specific objectives and outcomes, has been established. The inclusion-exclusion criteria were finalized through discussions in our group meetings. Choice of statistical methods to evaluate the imaging data is complete, and recruitment and retention strategies have been decided upon. The sample size needed for the study was reviewed during our small group sessions, and the final draft of the protocol is in final stages of preparation. The MRS approach proposed for the project is completely novel in assessing disease progression, and the course was instrumental in providing language in the rationale section of the protocol. The course was also very helpful in improving the design of the study to address specific objectives, which included primary, secondary, and exploratory components. Finally, given the exploratory nature of the project and the desire to study a rare disease, significant work was done in analyzing and discussing the sample size needed to answer the specific questions proposed in the objectives. Corinne, or am I saying your last name? How do you say it? Mal. Okay, I was going to say that. Well, that's the name. So what Corinne has learned in Ann Arbor, I like the way she wrote this, because we all learn things that's interesting to hear what other people learn. I need to change my overall objective of my clinical trial. Although efficacy is perhaps the most exciting objective, it is probably not the appropriate primary objective of my early proof of concept study. Number 2, much work needs to be placed in optimizing outcome measures and clinical rating scales of my disease of interest. Very true, and Mike talked about that Sunday night. And my favorite, that we shouldn't be afraid to fail. That I've learned that a failed intervention can actually be considered a success provided in clinical trials. Mallory. Mallory's project will test, so Mallory Hacker at Vanderbilt, studying Parkinson's. Mallory Hacker's project will test the hypothesis that a common functional polymorphism in BDNF adversely affects outcomes for medically treated Parkinson's disease patients. This week, Mallory has focused her study on early stage PD patients treated with standard medical therapy, which really focused the project down since we started talking about this a few months ago. Her recruitment for this prospective single-center observational study will be from the movement disorders clinic at Vanderbilt, and efforts dedicated to research staff to facilitate recruitment are being considered. Future areas of development include exploring the feasibility of subgroup analyses based on the prescribed Parkinson's. Mallory was also very good at writing the third person. Minish Ranjan at University of Toronto. Protocol framework was formulated while IND/IDE requirements and patient approval policies were reviewed. The updated plan is to suggest that the safety and efficacy of magnetic resonance guided ultrasound to medically refractory OCD patients and compare outcomes to the control arm. And the next plan is to update the proposal to draft the research program. 6.
So Michelle and I worked with four grade students in grade 6, and I think that the theme for many of them was realizing that, and many of us knew that the initial goal was to address it and stepping back and really thinking about what was accomplished. So I'll start with Mangaraj, who isn't here, who's interested in cognitive dysfunction and muscular dystrophy, and specifically in the hypothesis that working memory is a driver of cognitive dysfunction and potentially modifiable, which is the modification was the original goal, but we realized that we really didn't know much about the relationship between working memory and IQ and muscular dystrophy, or about how you actually measure working memory. So the project really is looking at this variable and trying to correlate these two relationships, and then stepping back towards the clinical trial when you get the biomarker and patient characterization defined.
So I will talk about Archana Ndua. I never pronounced her last name, so I said that wrong. I'm sorry. She's from Ohio State, and I think she's had a very enjoyable week talking to everyone and also learning about different ideas she had and how they may have resulted in a very large sample size or a large study that wasn't going to be feasible for herself. So she's been working through her objectives to come to something that's feasible in her single institution study. And I think where we are right now, and still investigating, and she's looking at moderate to severe stroke patients who receive TPA. And she's going to look at blood markers of the clotting system at 12 hours, and also evaluate DVTs at 5 days. So she's kind of getting the natural history of the patients getting TPA and looking at the relationship between these markers and DVT. And I think it's been a great progression watching her come to this observational study, and we'll learn some important things, and we'll get her to her next step of her ideas of where she wants to have her intervention. So I think she's really progressed, and I think she's enjoyed all the office hours and talking to lots of different experts that are available.
Ruth Sanju is interested in sudden unexpected death in epilepsy, and in particular the hypothesis that altered ventilatory response to a CO2 load potentially is predictive of SUDEP, and more importantly, if you can modify that, maybe can alter risk in SUDEP. So he has a tool which is called HCVR, which I don't actually remember what it actually means, but it's a way of assessing responses to CO2, and there's a bunch of different measures that you can derive from this test, and some evidence based on a psychiatric disease that both this response can be altered in a specific disease and also potentially modified by fluoxetine. So what he's doing for his initial project right now is, number one, characterizing this measure in patients with epilepsy, taking a wide swath of different kinds of epileptics and seeing whether different patients with epilepsy have differential sensitivity. looking at the measures that are available from this test, deciding which ones are the most reliable and the most sensitive, and then actually exploring the effect of fluoxetine on this ventilatory response and determining both retest reliability and the effect of fluoxetine to modify the various measures that are available with this test.
And our fourth trainee is Doug Rosell, and his project is therapeutic lumbar puncture in African children with cerebral malaria. And what he's wanting to look at is not only whether lumbar puncture can have a benefit in the death rate, but also desire to look at whether maybe multiple lumbar punctures are effective. And throughout the week, things have evolved as far as number of sites available and really considering the number of subjects you could recruit in a reasonable time frame with this grant, and the desire to have multiple arms in the study and kind of thinking of it as a dose-finding design, but then also with an assist from Kurt Bealey, we looked at some possible adaptive designs with the dose-finding to try to maximize the sample size, and maybe somehow being able to use some information from the historical data that he has. And as we were doing that, finding that there wasn't a lot of benefit to what he was going to be able to say at the end, and now has been looking at reducing it to a single arm versus control study where we can really answer the question, is there effectiveness of lumbar puncture? And maybe not looking at all the doses, but that would be the next step. And so hopefully that we'll still be working through that, but I think he got to see clinical trial simulation on the fly and see how that really works and how it can inform your design, and really looking at that the situation is the best way to answer your question, and sometimes the really cool designs that you may want to try may not be the best way to answer your question, which of course is a disappointment for me as a statistician. So I think we had a really fun week, and thanks to all four for working so hard, and I think they all made a lot of progress.
We had 4 people in my group, but it was 2 clinician-statistician pairs. So we really had 2 projects that we were working on. We had Mayor Patel from Vanderbilt, who was studying TBI, and Adam Havanon from the University of Utah, who was studying stroke. Both of them, fortuitously, are doing, while they're looking at different drugs, treating different diseases. There's a lot of similarity and a lot of synergy between their 2 trials. So both of them are going to be doing an adaptive dose-finding trial with a control and 3 different doses of their treatment. Mayor's outcome is the TBI version of the utility-weighted MRS. Adam is going to be looking at the effect of his drug on the change in the core volume of the stroke. That's the endpoint that's going to be used for adaptation. But the effect that they observe on that more intermediate endpoint is going to be used. We're going to correlate that also with the effect that we expect on the modified ranking scale. So there's been a lot of back and forth on the appropriate endpoints. There's been a lot of back and forth on the objectives for their grant, but I think when we got here, we settled all of that very quickly, because then the statisticians were tasked with sitting down and coding up simulations to simulate this Bayesian adaptive trial design. So we've been locked in the back room. You might not have noticed us, but we've been cooking up custom R code to be able to simulate their Bayesian adaptive trial designs. And the statisticians, to their credit, got pretty much entirely all the way through their code. So they're leaving here maybe with a little bit of debugging to do, and then they should be ready to fire it up and start showing everybody what the adaptive design can do and giving us some operating characteristics. I'm really looking forward to leaving this residential course, because then we can start in the calls we have after this
doing that interactive process of tuning the design between the clinicians and the scientists.
All right. So, Valerie and I, and to a certain extent, Chris, who we asked to join our group late, had group 8, and the theme of group 8 was sticking things in people's heads. And 3 neurosurgeons and also a movement disorders neurologist who did deep brain stimulation for Parkinson's disease. So some of this was. So I thought our group had made a lot of progress while we were here. I think the small groups in advance were very helpful to get to know the disease areas and to move things forward. I think so Kelly's project first, since Kelly was our REM Book Award winner. She is a pediatric neurosurgeon at the University of Virginia and interested in pediatric congenital hydrocephalus and looking at a new procedure, the endoscopic third ventriculostomy with choroid plexus coagulation as a treatment compared to the standard treatment of shunting. And we have gone back and forth many times as to what exactly the primary outcome of that study should be. As with any surgical trial, there are all the issues with blinding and recruitment, getting the treatment outside of the trial because it can't be done with sort of anyone. And I think being able to iterate and really coalesce back on, I think basically what she said she wanted to do when she did her elevator pitch after we moved her far away from that for a while. But I think that is reasonable in that, looking at how many people can you really get into a trial like this and how many people are getting the therapy that they're assigned to, because if you don't have that, then you don't really know how well this treatment is working, since it's a new treatment compared to a standard treatment that has potential benefit in limiting kids' exposure to having a shunt for the rest of their lives. So very important work, very, very tough work, and I think we've made progress while we're here. So the next project I will talk about is Louis Savastano. He is a resident here in neurosurgery at the University of Michigan, and he is interested in carotid artery disease, and this idea that for the stroke people in the room, this is pretty known that when there's bad stuff in your carotid, you may have a stroke, and that's a simplification. But the issue is that some of the bad stuff is stuff you can see on a CAT scan or a Doppler or an MRA, and then there's some stuff that you can't see, the plaques that are flat that aren't really messing with the lumen per se. So he has this interesting technology. He's worked with folks from GI here to develop doing angioscopy and actually sucking the blood out of the brain and looking through the artery with the camera. And they were already going to, so then this has been a good progression because we worked to try, who's going to consent to that study? Sucking the blood out of my brain? But we found some people that were going to be sucking the blood out of their brains anyway, so not the brain, the artery. He's cringing. He's like, what? All right. So I think we've made a lot of progress there. I think that's interesting device trial. Again, there's going to be a big focus on feasibility and getting usable images, sort of dialing back from the sort of initial idea of let's give maximal medical management when we find things. We really need to know how we can find things first. So that, I think we've had great progression there. Unless you have any other thoughts. So next we have the Unreal Donald Trump from the University of Minnesota. David there. He is interested in refractory epilepsy, but mainly in using low energy focused ultrasound to target areas to reduce seizure frequency. And this is a tough problem. We had a project that was a little analogous to this before, where it's a big challenge because there are people who seize 20 times a day, there are people who seize once a month. Both groups would be considered refractory epilepsy, but if you're doing a trial to look at a therapeutic modality to reduce that, those are very different trials. And setting up a trial that can reduce somebody's monthly seizure to less than monthly is a little different than reducing somebody's 20 seizures a day. And I think we've gone sort of back and forth, and at one point we were really I was pretty ready to sort of throw in the towel and say we just need a cool Bayesian adaptive design at 1,000 to 10,000 rats, and then we'll be able to really like learn something. But I think that there's also very important stuff to learn in the humans. Again, this is a little bit of a idea to sort of learn in advance whether the patient's going to be a candidate for an operation to more definitively manage their refractory epilepsy. So that is, so I think we've made progress, and I think we've reduced the parameter space. And I think once there's a little bit more of this preliminary work done, I think there's a very potentially serviceable design that will answer the question at sort of high doses, high durations, but in a proof of concept. And finally, Drew Kern from the University of Colorado, studying Parkinson's disease and deep brain stimulation. The idea of his work has been that usually leads are placed in the subthalamic nucleus. On your way to the subthalamic nucleus, you're going through the rostral zone of incerta, and that is a place that it is potentially good to leave a electrode lead. Stimulation there may be just as good or close to as good as the subthalamic nucleus, but you don't have to just settle for one because there can be leads in both places because of the way that the technology works. And potentially co-stimulating both regions can lead to a better side effect profile and equal efficacy. So a lot of what we focused on there was learning about how often we can really get into that area and know that that's what we're stimulating. And I think that's important because if you're not in the area or close enough to the area for the electrical current to penetrate it in a biologically meaningful way, then that's not really the intervention that's occurring. So I think we've made great progress. I think they have good plans, and we look forward to seeing them do great things. All right, let's give Group 8 a hand.
I will describe the projects because there's no way I could have done that. I think it took me this week to figure out how to pronounce angioscopy. But what I did want to say about our group is that I think we had, even though everyone had very different projects, as Will described, the common theme was we're in this early phase of feasibility. And how do you write a grant where one of your primary questions is, are people even going to consent to be randomized? And how do you make it attractive? What kind of funding mechanisms do you use to answer these early questions before you actually jump into it? So the other thing is, how do you justify your sample size if you have outcomes such as the feasibility of getting into the zones or the feasibility of people consenting and committing to what they were randomized to? So I'm sure others in this room have probably been in the same position, but so it'll be interesting to see how these move forward.
I guess I didn't give any humorous anecdotes from our group. So since we have 3 neurosurgeons and I'm an emergency physician, sometimes I didn't manage the time very well for our group. So we spent a lot of time talking about somebody's project. I'm like, oh crap, we need to finish this in 5 minutes because I'm hungry and need to go to lunch. So at times it was like, okay, think epidural, hematoma, get it, burrow right now, right? Let's move this on. So a couple other things before we break back into the small groups. Okay. Oh, we have group 9. Sorry, I forgot group 9. We had 9 groups.
What were you saying about managing time?
Epidural hematoma. I don't think that works for biostatisticians, right? You don't take out epidural hematomas?
Okay, so group 9 we have a big group, and Rob, Supergay, and myself are the group leader. I didn't see Rob around, and but I think the good thing we have a good group. The important thing is trainees evaluation, hardworking for the last few days, and I think we're making good progress. And as a general metrics of our progress, I have to say that in my usual confused state of mind, I actually managed to remember each one of the studies design, endpoint, and the patient population. This is a big deal for like a personality, so I think so actually, so they work really hard, and I think they are working hard in each of the protocol. So to give them credit, and I think, and actually I think we are ahead of time. So I think to ask each one of them to talk about their own project. So I sort of take advantage of the fact that we're the last group and best treatment. And so let me call upon each one of them and so that they talk about. Do instead of 5 minutes elevator pitch, do 1 minute. You need to so first, Brian Kendall from MGH.
Hi everyone. I study traumatic brain injury, specifically looking at imaging and EEG-based biomarkers to predict recovery. And the trial that I'm proposing for this course is one in which we would test an acute stimulant therapy to promote recovery of consciousness in the ICU. Pardon me. The outcome biomarkers that we would use from a clinical standpoint are the coma recovery scale, which is the gold standard for measuring behavioral or clinical improvement. But I would also be testing the impact of this acute stimulant therapy on the biomarkers that our lab has been developing and testing for the last couple of years, specifically MRI-based resting state functional connectivity and EEG-based cortical network connectivity. The main lesson that I've taken away from the last few days is that whereas I started with a design that was a phase 2 placebo-controlled randomized crossover design study, I realized that we need a lot more information about the pharmacokinetics and pharmacodynamics of the stimulant that we aim to test, and therefore I'm going to move backwards, so to speak. Hopefully it'll be one step back, 2 steps forward, to a phase 1 study where we look for both pharmacodynamic, pharmacokinetic data, as well as performing a dose-finding mission where we look at different doses of stimulant to look at clinical responses and biomarker responses.
Great, thanks, Brian. And so our second trainee, it's also like a neurocritical care physician, Holly Kingston from Oregon.
So maybe they grouped us all together. So, like Brian, I study severe acute traumatic brain injury and really interested in progressive cerebral edema and trying to predict that in order to sort of thoughtfully include patients into a phase 2 open label study of glyburide, which is a sorbontera P4 channel blocker that has been demonstrated in acute stroke to reduce the line shift, thinking being by reducing cerebral edema. And we're hoping to try to very thoughtfully translate that into TPI. And so this week, I've really just been working on my protocol and kind of trying to trudge forward. So,
and then next we have Kamal Sahadi, from Irvine, and talking about studying about coherence.
Hello everyone. So yes, my study and my work is studying the efficacy and tolerability of the pyramid in controlling abnormality behavior in behavioral areas. So I knew that I was in rarity, but now I just I've got the unwanted child as well. So like everyone else, I mean, I was a bit back and forth this week, and it was a reality check for me as well. My main challenge was to decide on my primary outcome measure, given the rarity of my disease and the fact that there are not many validated methods to sort of assess it. And I have been really back and forth with this, but I think I have settled on the fact that I think we do have a validated measure, and if this is even any use to me, so, and the other thing was to decide on a crossover design as opposed to my initial thought of having sort of placebo control. We decided, given the rates of disease again, it's good to go ahead with crossover design, but I'm aware of all the sort of challenges. So I think in summary, it has been a great week for me too, and I learned a lot. The most important thing was to not to try to neglect the potential problems that the design might have and try to tackle them. Great.
And last but not least, we have Charlotte Sherner from Calgary. And I heard that there's a fantastic design going on there.
Yeah. Hi everyone, my name is Charlie. I'm one of the stroke fellows in Calgary, so naturally my studies are acute stroke. Specifically, I want to test the earliest, safest time point to initiate oral anticoagulation in cardiomyopathic stroke. the risk of recurrence is highest in the first few days, and there have been not many studies on this, so this week we're going to spend a lot of time talking through different adaptive designs, and then finally, I think yesterday deciding on one, how to push forward as much as possible, but not getting too much information about the days we already know about, but the early ones we do not know about. Maybe even kind of going with a smaller sample size than initially anticipated, so potentially now as a single center study.
Great, thank you. And thank you.
So before we go back to the small groups, and one of the ideas of these small groups is to get a sense as to what people should be working on, and then Sam, we will be working to help the groups go back to the scheduled office hours for the small groups with the 3 sessions that remain. We have some webinars planned as well. One other thing, since I'm standing here and I have the microphone, I want to put in a plug for the Society for Clinical Trials. I am one of the co-chairs of the Scientific Program Committee for the 2017 meeting, which is in Liverpool, which is in England. While it is a far trip, if you haven't thought of joining the Society of Clinical Trials, and I will say that when I was in your sort of station in life, at least for the clinicians in the audience, I hadn't, but it's a great organization. And a lot of times when you're like, I need to find somebody who does this, there are a lot of people there. There are a lot of people there, like some of the world experts in step wedge designs. There's a whole session on the step wedge designs. It's a great opportunity to collaborate, and even some of the designs and things that you're working on might be interesting as poster presentations or even perhaps oral abstracts. You don't present primary clinical trial data, but a lot of the nuts and bolts of doing clinical trials, whole sessions on the surgical trials. I mean, this has come up a lot in our group. How do you do these things in a good way? And some of the worldwide thought leaders in how to do these things, and plenty of statisticians there, graduate students who are doing interesting new designs and can potentially help you with things. And you could potentially get an international presentation out of it if you want to go to Liverpool, and you can see me there. So that would be nice. So so after we have our small group here, we'll be coming back here for sort of 2 main more didactic sessions. We have a little time also dedicated to you filling out evaluations. Again, we want you to live up to those 11 to 100 surveys that you will fill out during the course of your participation in the course. Just to sort of look at the number of people that we've touched so far in the course, and maybe to give you all a little bit of motivation. Again, we're getting close to your protocols being finished. You're also going to be hopefully preparing grant proposals, and again, if you're using an NIH mechanism, it's pretty straightforward. If you're using another mechanism, you don't need to translate it into the NIH mechanism for our course. Put it in the AHA type form and turn it in for our course. But we will be doing a mock study section, and your protocol and proposal needs to be in by October 15th. So one of the things I can say is again, we're all competitive people, so let's just talk a little bit about how things have gone before. In 2014, we had 23 trainees, 7 participated in the mock IRB, and 6 participated in the mock study section. So that's about 25%. They weren't mutually exclusive, some participated in both. In 2015, we had 36 folks in the foundations track and 11 folks in the advanced tracks across 4 projects. We had 22 participate in the mock IRB, meaning they got a protocol done, and then 9 participated in the mock study section. And again, there was overlap. Most people who participated in the mock IRB also participated in the mock study section, although there was one situation where that wasn't the case. So that's better, like 25% the first year, like 66% got to something. So for 2016, we're not going to have separate mock IRBs and study sections. We're going to have a study section that's going to address human subject protection similar to what a real NIH study section would, but to really be focused on the science and giving you feedback on the proposal. And again, the best way to describe the science in more detail than what you can fit in the 12 pages is through the full protocol. So we are really looking forward to a greater number than 66% getting to that. And again, we need the documents in by October 15th so that we can start assigning those to groups. And again, we'll be making sure that people who are, depending on the level of expertise, we may have some faculty who are both from within the course and perhaps some people who are from outside the course who will be doing the reviews. But these reviews will be being done by people who weren't involved in your small groups before. We had a lot of that mixture while we're here, so I know that you had office hours with a lot of people. So it may be somebody who saw your project before. I can't guarantee it won't. But it will definitely not be your small group leaders who are going to be the primary and secondary reviewers of your grants for the mock study sections. So we're really looking forward to the progress there. And again, the 2016 cohort is going to beat the 2015 cohort and the 2014 cohort. Although, Adam and Mayur are going to be like sort of beating themselves by getting their protocols done. All right, so we have webinars around September and October. One of the ones in October is, they're all going to be great. Some of them they're going to be a little bit more tailored towards grant proposals. We're budgeting for 3 small group meetings over this time period with your proposal due October 15th. The mock study section will be in a November date. And in sort of contrast to a regular study section, we are going to schedule this based on the faculty's availability, but we are going to record it. We're not going to post it on YouTube like some of the other things, because obviously it has your own specific projects that we wouldn't want to broadcast to the world. But unlike a real study section, if you are able and can attend during the time we're discussing it, you can come watch. And that is something that you don't get to do in real life, but hopefully would open your eyes to the process a little bit. So again, keep the red proposal due October 15th. That is sort of the prerequisite. The American Academy of Neurology has graciously supported aspects of our conference, including our dinner, and they graciously support a reunion each year at the annual meeting of the American Academy of Neurology. And this next year is going to be in Boston. We have people from past years present their updates. And again, we actually focus on the last year's cohort the most. So for those of you who are going, we'd like to hear from you, and we'd like you to give us perhaps a couple slides and a brief talk about where things have gone with your project. And you can also see where things have gone with projects from people from the earlier cohorts. In addition, we will send you the full number, but if you have publications that are on work that was helped to be developed by this course, it's helpful to us and also to the Institute for you to reference this R25 number as supporting your science in terms of if we want to keep this project or a project like this going over time. Being able to measure the impact of the project through the publications you're doing will be very helpful. And I think in terms of completing things, the sort of, once we get through everything by November in the mock study section, we will be giving you a certificate based on these following criteria. The points that you got in the knowledge test yesterday do not change you within these categories. You attended the course, which is good. I think there's a lot to learn if you just attend the course. But as this is a project-based course, we want you to have submitted a protocol and a consent. This is for the situation that you're like, yeah, I couldn't get to finishing the proposal, but I at least wrote the whole protocol and the consent form. Perhaps you're in a situation where your department just has a lot of resources and soft money, and you don't even need to submit a grant. And this is then you've completed it. But we would probably give you credit on the third category, completion with distinction, if you're actually already enrolling in your trial because you have funds from your department. But submitted a protocol, consent, and a proposal and participated in the mock study section. So that's the highest level of certificate that we will be providing to you at the end. With that, questions, Chris?
I was going to add one additional point. The announcements that this is the third year of the course, but we're still trying to increase awareness of the course and get more people who are potentially, could benefit from the course involved in the course. And so Will, Lori, Robin, and I can talk to people, but it's different when you're the person organizing the course and you're telling someone. You guys, so if you didn't like the course, tell us about it on the evaluations, right? But if you did like the course, let others within your institution, other colleagues or friends that do it, because in a lot of ways, you guys are much more in touch with the cohort of potential applicants for the course than we are, right? It's all your friends and colleagues and people that you're working with. So if you like the course, tell your friends. If you know someone that you're working with that thinks they want to do clinical research and you think you got something out of it, tell them. Tell your chair if you liked it. Tell others so that they can encourage someone who might not be aware of the course but is in a very similar situation to what you are. So whatever. If you didn't like the course, tell us about it. You don't have to tell other people. But if you did like the course or you think you got something out of the course, tell your friends and colleagues, because again, you guys can reach more people probably who are potential applicants for the course than we can.
Yeah, no, definitely. I would say also, particularly if you have friends, colleagues, people you did training with who are outside the sphere of sites within StrokeNet and NeuroNext, I think those are also very important groups that we want to bring in to see here. I think we do a pretty decent job of letting the people know who are within those networks and the Neurologic Emergency Treatment Trials Network because of Chris and Lori and our Valerie, the faculty from MUSC on NET and NeuroNext and StrokeNet. But I think if you have colleagues outside of those areas, definitely letting them know. And again, the other sort of thing that sometimes people ask is this, what counts as a brain disease? And I think that's something that they can always feel free to talk to me about. We've had projects that are more from the psychiatry fields, more from rehabilitation, even things that are more related to vision. And I think the optic nerve to me counts. And there's the rest of there's the rest of the nervous system, including the peripheral nervous system. So it doesn't have to be it has to be neurologic, neuroscience. But we keep a pretty big tent with respect to that. So I think about whether they can benefit from the course. So with that, let's break into our small groups. We'll need to be back here at 10. Is that right? Yeah. All right.
Well, I haven't unpacked this stuff. Yeah, you should be that. Is Rob here? What are the dates?
There's a decent chance we'll see you guys again before you have
Okay, cool. Either way,
Thank you so much. See you around, okay?
Yes. It has been very successful.
yeah.
be honest, I can't remember because I'm originally I'm trained in the UK. That's why. There, I mean, there's before now. Anecdotal evidence.
Oh, they weren't very
It's promising, I think. I mean, what we'll see.
All right, well, I'll catch up with you later. You know, as a beginner.
Yeah, I want to say that.
I think putting it, sort of plotting it out is like.
No, I haven't.
This is what I'm
It actually, it really did help me a lot. I can imagine. It really did, like I found myself like, actually,
we want to give you some feedback. Strong case. So that's going to be done. So I think that here's where
yeah, so I was going to come home. I would just work on it Oh, at 10? Not too much. Oh my God, I'm starting to crank. I might just take part of it. I know, I'm telling you. You really need to get a new one. You need to. I'm sure it's at 10. Oh, it's at 10. You have breaks and okay. So, and then it's this and I was curious about the 10:45 and not much.
Well, I think as many ways as you can include.
Unless there's more.
Because part of it is, if you're writing an R1, you only have 12 pages to put stuff. Other things that you can put into the subject section, like emphasize justification, things like that. That whole sort of process of like looking at these through the instructions on those, that I think at the minimum it needs to have the research plan, but the more of those other things that you put in, the more people will be able to sort of, it should have a human subject protection section, just from the perspective so that we can see where, because that's part, in a career review process, that's what basically, it doesn't go to the major scientific score, but it has to be, so aims, research strategy. Yeah, we'll make that more clear. At the minimum, it has to have the aims, research strategy, the subject section, and then, ideally, as many of the other sections. Is it okay if I was to this is a lot? Is it okay if I use your homework? Yeah, don't submit the book, don't submit the protocol for the trial that you're not doing yet, but it's okay to have, but this is the phase 1 protocol, just building a little that would say that we would adjust this.
I just felt like I needed to come back. I felt like I needed to
I appreciate it. Yeah.
so, I just felt like when you're it's not tell him that. So how do you explain? I did, she knows. She knows. The only thing she did was she just allowed her kids without charging. Because remember, there's a hard cancellation. She's so I was like, I appreciate it. I was like, if I do start, like in a classroom, I'll just yeah. In the meantime, I thought, so this one's like a slow, heavy, like, maybe we'll go down until like 20, move through. Move it. And so, there's so long, like maybe the weeds that I want, but maybe you can do like farmers. That's what I thought. There's so many farmers. I just never feel motivated to do it.
I know, but I think, well, at least for me, if I haven't done it in a while, I'm motivated.
I know.
I just feel like this week especially.
What? So to get me stronger, God, that was here. Down. And down. Just. I don't know what's I feel like the ones that pass these, and it'll go away, but. Morning, morning. In the school. Are you guys here for some? I'm excited. Blue jeans work out?
Are we all getting the experts for that?
Oh, they're good. Oh, good.
Are we all getting like an early happy hour drink after this? Yes. You want to?
Oh.
You're exempt from that.
I was eating a croissant. I know that. It's like flaky
Croissant
croissant.
was good.
I hear something, but it's very faint.
That green one. The one driver. Just don't worry about it.
Hopefully they ran some chemical tests on it.
They did, but it was still, it's just, it happened so quickly that I don't know about that. I'm sure they wouldn't have wanted to put it in
Put
there.
something in it. I don't know. Did somebody put some dye in it or
No,
something?
it was because it's an outdoor pool. It's so warm in there. It's like a really warm day. And the algae just, their filter wasn't working or something. And the algae just produced like immediately. And it got greener as the
It got thicker and thicker.
It was gross. But they couldn't see the bottom of the pool, which they were always used to seeing, like, it being so clear. And it can affect your diet, but apparently it didn't, so.
We were saying that about a couple of days ago when we first started, just the weather was so windy and it was out we were outside. And I'm like, some schools I guess you can't control the weather. And I guess a lot of winter sports are like that. But I was down at Salas.
So they had their orders. We basically had chicken kebabs. Never tried it. That was really tender, but juicy. And then they had that is the how do you say the meat plate and cheese plate. It's not crudité. Crudité is vegetables, but this is like awesome cheese, salami, it's really good. And the souffle, yeah, like, oh my god, it's so good.
Was that like an appetizer?
Was, yeah, it was just and for dessert, they had you want to try the little cup, shot glass of water. Like they had like a peanut pie and like a fruit, mixed fruit, like pork,
Diana Taurasi
and then
hits her 6th 3 of the game, and her Olympic record.
Yeah, but upstairs is the bus. You know how they have that long, way that tables up against the back?
You need a website.
None of that
Why not
was
do it
done.
yourself?
And then we had the back.
Easy to create your own
And I
site
guess the
website.
background.
Browse hundreds of beautiful tables. Choose the one you like and get started.
Or an area of business and stuff.
Simply enter your business name.
Yeah, it was just it was a little loud, and we were the front.
Change your
people.
background.
So like one of the speakers had the
With a
best
click of a
voice.
button, you can customize anything you see.
We're scoot back, like I can set up the bars that we can hear.
Introducing
So it
That was
was a
pretty
little hard to
during dinner.
It was after dinner. It was like every now and then, but it was it was a little loud. Yeah, still. I mean, it wasn't crowded, but it was still the acoustics in there. So it's loud in there anyway. Yeah. The food was okay. I mean, I think I've had better. Yeah, it's families like that. It's just,
I don't know. I didn't want to say anything because I've been hearing it. So then I just let you guys go and see what your guys' experience
I was
was.
telling her like other places that have like large rooms. I was thinking like Montreal has like a side room.
That's right. The Belleville has something downstairs, right?
They have a room.
Oh, it's not that cabin spot. Yeah, big enough.
Yeah. And it's awkward because it's long, so it's not like it's hard to just.
Because you could have rented out the bottom. I mean, I don't know, maybe
The whole body, yeah.
oh, it's right.
Yeah. So I said maybe for another year, check out Black Pearl, and then, and she was saying like Poly on the rooftop, but I guess Main Street's a little far from me, but
I mean, it's actually wouldn't be bad if they stayed in Marriott. They stayed in Marriott. They're always on that. Well, even here it's just it's not far.
it's like 4 blocks. It's really not.
Especially with the weather going to it's been nice.
It's got to be like 4 blocks, right?
Joy?
We
You hear the other, oh.
Yes. Hello. Oh, so, oh, that's right.
For questions, people ask during the conversation.
Get up. Yeah, they should. People aren't gonna go. So I was trying to think of other restaurants that have space.
Well, Lisa Lita's got that space downstairs too. They have some private, but I don't know if it's big enough. I was just there last night. I went to the bathroom. I was thinking that reception, but honestly, I don't know if it's big enough.
I think last year they went to.
I did the bubble room last year. I did the bubble room at biology.
Biology. Same thing here, but biology. It was a big.
Yeah.
I've never been there, but
It was
that's what it was grand.
It was, and that seemed like a good space.
I know people like want to sit down, but
Oh, that's right.
still, it was.
I know she didn't do a Marriott because of the, even though they have the rooms for some people who don't have the space or something like that. Which is too bad. Yeah, that airline. Yeah. So
I love this, and I think people really enjoy this. And this is me by our too, but like this is
yeah, because immediate access to campus if they want to walk on campus.
Right.
And then
There's close like the CVS is just in walking distance. There's.
I saw people the other day when I was here had carried had like zero disputes on like what their carry time. They must have walked the carry time. I didn't know if maybe they just
It's frustrating. It's from a budgetary. People don't really think like when you're at a con, like 10 minutes or on other invites, I'll go. But really, they really because like chaos. Which I didn't really realize. Like here in Minnesota, there's 4 cans of soda, and so there was some extra. So the guy was asking, "Oh, you want us to return them? And we just want to charge for those or not?" Like, so you just don't think about it and stuff like that. It was like I was ready to go after that. I was like, it's been like 10-hour days. I got out a little early yesterday, which is nice. Because I didn't have to, it was like small groups and then
initially, and so I said, she goes, I mean, she's like, what's that? Like, what goes on? Because of her hours. So then just don't work on Friday. I told her flex it. Yeah. I said, can't she flex it? She said, that was said, she made such a big deal about that. I don't work. And I said, why can't you just flex it? 'Cause I'm pretty sure that the hourly people can flex their time. Pretty sure. I know. I just don't know. I don't know, but I'm weird about so many hours in Jenna. Well, that's what I said. I think I heard Jenna once say that she flexed her time or something because she had an appointment early in the morning, so she came in later and stayed later. I said, why don't we ask her to tell it? Just don't get to get I said, I don't think Robert's going to tell when she was I'm not going to say anything.
That keeps evolving.
So wait till they're back. And I said, just don't come tomorrow or come later tomorrow or whatever. She's like, apparently
You took out all this,
speed. I don't understand why I guess she just doesn't showing us that anyway. I think that right. Well, Lindsay said, well, I'm not going to clean it. Nobody said, well, then just come for dinner. Like, you gotta eat dinner, right? So, yeah, come for dinner. So, and I don't know if she's got, like, friends he had around here. I know some other people, so I think, like, how much of it is, like, you'd appreciate it. Yeah. Well, I know, because, Lindsay asked about, there was, like, if she needed to check in with him about her schedule or something this week, and she's like, well, she was thinking no, but it's that. She was like, well, what are you thinking?
you're talking about dinner and how
No.
proud I was about taking me to dinner because I'm going to hear about it.
No.
I know I will.
No, you're not.
Robert was there.
So what? Robert was like,
Somebody was saying something.
those were my exact words. Well, and the thing is, Val just, she said, yeah, you know, I just told Lindsay, I don't take cabs. Sam needs her, Sam can, I said, she's like, Sam,
I'm just saying that Val and I had a very heated conversation.
we did, it's so weird, because at the end of the day, last Friday, she was really awesome.
Well, I mean, so I think it was because of that conversation. And granted, it was, you know, early on when I first came back from India. She was just, I think, struggling with generally being listening. So she may have reacted. And there was like, I'm not dictating her time or people's hours. It's not what I'm doing.
Well, the thing is, like, she allowed me to do she couldn't falter to me.
Exactly. I was told that our can't flex time. So
But she told us in there that
I'm just
Val said.
see, Val didn't say that to me. She did say if you need help, but she didn't say, you know, if Lindsay ends up working 12 hours one day, 10 hours the next, she would have to come in. You know what I mean? That
Of course she didn't have it.
But if-
And
but if it happens,
plus, she had an hour. Okay, so this is it, because we figured it out when we were in nursing. Remember that she worked 7 hours, so she had 1 more hour to go. So that's her dinner.
just saying, you guys, Val's in the
I will
hallway
support you,
shaking
okay?
her coffee like she's been smashing my face, and she's so mad.
Okay, you call me in and I'll take care
You
of it.
know how she is.
I do, and I'll take care of it.
Well, she's in the
What, are we live? Are we live?
Yes. What does it say? You are the first.
Like, what time is she? She's in Georgia. Oh. Sweet phones. I don't think she can hear us here, honestly.
This guy jacked up the sound system.
That's David. All right. Well, we know she doesn't have the link to get it, so. Yeah, so I guess.
And seriously, I was thinking over and say, this is how it was. She didn't come in the other day.
Well, I didn't think about.
What's
like,
happening
that.
now?
What? B. Willis.
B. Willis.
I didn't think about like if there was everything to your day. They think, you know, you're there past your,
Right.
you know, when you were playing. What is it? What am I gonna do? Robert? Yeah. Sorry, my time's up. I'm just like, it doesn't even make any sense. It was 15, 30 minutes, whatever. You know, I mean,
So come in 15, 30 minutes late. And that's what I don't get.
she makes us look like if it's that time, then people flex around. She's not working all the 40 hours a week. That was my whole thing. I told her, just thought you were doing it. And he said, Thursdays. And I was like, I thought she was going to be years.
Right now,
Then she's in flip-flops though, so like she tells me one thing, she tells someone else another, you know what I mean?
Yeah, I was being quiet.
There you go. Hey.
Good morning, how are you?
Good.
Yeah, I can hear you.
Okay. Is your camera?
I was trying to turn it on. Good morning.
Okay.
I originally had it turned off because I wasn't sure where we were.
Oh, so we're in the conference room, but it's empty right now. The next probably 10 minutes or so.
Okay.
The angle is awkward.
Yeah.
The way she said it was.
And then do you want to try the
Yeah.
slides? Sharing your slides?
Let's do that now.
And maybe moving the camera to probably your left to my right. Because right now you see this door.
Yeah, I did that.
Yeah, but
Maybe we don't need some video actually because it's
it like is it a better sound just so, no, it's just a similar conversation I had with
Can you see my slides? Sam, pardon me? Yes, you know, it is change for you. Okay, so what I do is, okay, you can make it right.
It's almost like you're ready
Yeah, I shifted my, so I can, usually be like this. Is that what you like?
Translated to.
That's right. And I think now I'm kind of
It sort of like validates or not sort of. Yeah, I sort of anticipate.
yeah. Okay, you're
Yeah.
I know you're trying to do it, then also that they have all these template and like sending it out. You just send it out.
Yeah, like that.
Yeah.
It was built in 1938, like there are things that need to be done. Right. You know, I think that's good. I have to can't even. It's ridiculous. That's right. Like, there's like 3 different problems.
Okay.
Are you okay?
The way the gray and the white.
Oh, thank you. I know, it helped.
He needs it. Just in case he's
Yeah,
out.
holding pattern?
Yeah.
Okay.
Efficient use of tell.
I mean,
Right. Yeah, of course.
Give her something on the table, and something on the table falls on you. You sit there and you look like you're getting the update for the first time too.
Yeah, I wasn't having any trouble with knowing like which slides were coming up. Now it's trying to do some help.
You can uncheck this, sorry. I was just trying to make sure that you can see it like full view.
Well, when I do this, do you see full view?
1 second. Yep, that's perfect.
Yeah, okay, that's what I'm most comfortable with.
Okay, yeah, it's perfect now, all right.
Okay.
So we're trying,
Okay.
I'm
Did I
actually,
have,
oh, go ahead.
I'm sorry, so go ahead, please.
So I'm running the AV today, and trying to see if we can patch it through so people can ask you questions through the actual microphone. Right now, we're just asking questions through the laptop. So
Really? I give that impression. She goes,
No, I think, I'm
maybe they're 2
comfortable
o'clock.
with it.
And I'm like, okay, well, yeah, I guess I do feel that way sometimes.
Finally got on, I had to wait because
There's a lot of inefficiencies going on, or inefficient conversations.
she's like 3 hours late on
So,
Chicago flight. So they were fine.
see that, I guess.
And
I
now,
said,
you know, with
so,
her take off, what happens, take your seats, she watches like the plane takes off, she takes off, they're up there, so they don't turn to the runway, they turn the other way.
oh, they did, yeah, it was like,
So by this time, it was like 1 a.m., she said she went over to the hill, when she got there, they were like,
yeah, which I totally appreciated, and I was like, and I could totally
so she
see, I
stayed there
that,
till
like,
they kicked everybody out.
some of our stuff, like, yeah, and especially like her involved and everything, but yet I'm not. So, we're good, but I'm now just about it, so I got Carolina, yeah, she does, but she translates it. I mean, she's smart. I can see that. Be able to anticipate or go, oh, something like that. I know exactly how do I see in my head already kind of thing. I feel like that seems to be good for her, and so she's just kind of you know what I mean? Like I feel like a little timid approaching her to ask her for something. But I feel like as far as her abilities she's bright. I think she's up there with what Allison was in that search.
I don't know.
Yeah, I feel like she is.
But, you know, I think it's too late for this year.
I know she was trying to
I think it was no preference.
but Robert
It's been totally decided, yes, but
there was opportunity to write, and she's actually interested in writing grants and stuff like that. She ultimately wants
the hotel, like,
so she gets her but she can't get it.
traveling internationally, you're going to over, so they'll charge twice for the day rate.
She's young. She's not over. She was really having a hard time finding a job, and so she's like, yeah, I'm just kidding. I don't care where.
Yeah, they got a lot of international travel.
Yeah, and she's young enough for She's like, another state. Yeah. Make it work. Yeah, she's good. Yeah, but yeah, she's definitely she decided like she just doesn't feel like apparently we're not. I'm just too. I think sort of like or like the most I possibly something like that. Surfaces things. So I think she would teach or something.
this afternoon. I said, since you have access to our supply group, maybe do you have any cancels to she's absolutely this party this week. No, I know. There was a conflict, but you guys were like, oh, I'm doing this, and it's like, waiting for us to come after. Sure. That's why I thought it was awesome. You left in the office.
Waiting's already been.
Tuesday, before I came here. You popped in just to I saw him for a song. He was he didn't come in the office. Stay as far as possible.
Because it's not like he's having it, he's having to do, not like our jobs. Where are you going to go? You go to house in those conference rooms. Yes, that's true. You learn that was in Candy, the last one, the right, because that's where there's some dollars.
Yeah, it's all right. Oh, come out, go to right?
No. Come out of here and go to the left.
It's all right. Okay. Across from like the gas station or something. As soon as we meet you, right? All right, I'll check on my way to the back.
And then, so they always do.
That's right.
Just don't, like, if you shut the door, put the bolts on, because
Oh my God.
so just put the bolts on, so it just hits the.
Snow White and the Witch was like the story of the circle. So this year, the theme is tomorrow.
Oh, is she?
The theme this year is down to our track. They have slot.
Oh my gosh.
Because
Lori's gonna say that.
Did you go to that, Sam?
It's on the topics and things we're going to do next time.
You know, Lori's going tomorrow, whenever Francis takes a slot in her. We have that little
you doing the Q&A?
Yeah.
I'm trying to work it out right now. I wasn't aware that configurations were different. So you can ask questions through the mic right now. You can't really ask them through the laptop.
Oh, okay.
So if I'm going to find out, hopefully it's going to check and see if I have one, but it's not here. So it'll take me too long to run and get it. But I mean, we tested it through the laptop, works fine. So we've had these that come up before where the moderator will just repeat the question. It works fine. And I checked, and the gal that's on the other end of the just doing the presentation, she can hear us perfectly on the laptop. So we'll be able to do that. So instead of having a mic, maybe you just go, okay, this person's next. This, you know.
Okay, yeah. Okay.
So.
Well, then I won't really need that then.
Yeah, well, maybe. We'll wait.
If the moderator can hear their questions.
That's what we'll do. Well, they can still ask it through the mic, I guess, but the moderator would just repeat the question after. But she may be able to pick it up off the mic. We've had that too.
Okay. So we'll just have to. Okay.
Sorry to give you a heads up. So
Thank you.
what's going on?
No, that's all right. Thank you.